Next, I'm honoured to pass the stage to Dr. Stefan Hauser. Dr. Hauser is the head of the Stem Cell Laboratory in the German Centre for Neurodegenerative Diseases in Tübingen. He's working on stem cell models of rare neurological disorders. Dr. Hauser has tested the smart lipid droplet assay with neurons. So, the stage is yours, Dr. Hauser.
Thank you for the introduction. Our lab at the DZNE in Tübingen is mainly focusing on disease modeling of rare monogenetic neurological disorders by using iPSC-derived neurons and other neural cell types. The two main diseases we are currently interested in are hereditary spastic paraplegia, HSP, which is an upper motor neuron disorder, and spinocerebellar ataxia, SCARS. We believe that lipid composition and metabolism can play a crucial role in many neurological disorders, and this is why we are keen and especially interested for potential in vitro-based readouts. One way to analyze impaired lipid homeostasis is to assess lipid droplets. This is why we were interested in establishing a lipid droplet assay in our lab. The NanoLife system allows to analyze the dynamics of lipid droplets in your cell type of interest in high resolution and label-free. This is why we are keen to perform the alpha testing of the newly developed software to quantify lipid droplets. On the right side, you can see an iPSC-derived neuron already cultivated for several weeks in our lab and then transferred to the NanoLife microscope. As lipid droplets have a specific and high refractive index, you can nicely visualize and track these over time. During the alpha testing, we performed some analysis on iPSC-derived neurons as well as astrocytes. On this slide, you can see a video of an iPSC-derived astrocyte treated with 50 micromolar oleic acid to induce the formation of lipid droplets. We performed a live cell imaging over 10 hours with an image frequency of 1 frame every 20 seconds. The software now calculates the mean lipid droplet count over time, or for example, the mean lipid droplet distance to the center of the cell. What you can see from this analysis is that oleic acid treatment induces the formation of lipid droplets in these astrocytes, while the lipid droplets stay close to the nucleus and are not transferred to the periphery of the cell. Here is another example of an analysis we performed during the alpha testing. In this case, we used wild-type iPSC-derived cortical neurons and compared it to a neuron where we introduced a specific knockout mutation using the CRISPR-Cas9 system. We believe that this knockout should result into an impaired lipid homeostasis. And indeed, what you can see that the induction with oleic acid results in a massive initial increase of lipid droplets, which then decreases back to the initial baseline values in the wild-type neurons. In the knockout neurons, there's a much lower, weaker initial response to the oleic acid treatment, but a prolonged treatment effect as levels are not decreasing over time. What you can also quantify with this software is the lipid droplet area over time. This, for example, allows you to see the effect on the lipid droplet size upon treatments or when comparing disease to wild-type cells, as in this approach. To convince ourselves that what we are quantifying are indeed lipid droplets, we performed a standard BodyP staining at the endpoint of our live cell imaging. What you can see over here is that the lipid droplet analysis segmentation tool from the NanoLife software nicely correlates with the classical BodyP staining. I really believe that the NanoLife microscope together with the lipid droplet analysis tool is a great option to analyze dynamics of lipids in a natural environment, as there is no need to add any labels or staining procedures which probably interfere with cellular processes. The software is easy to use, and we are really looking forward to future experiments to further investigate impaired lipid homeostasis as a potential disease mechanism in several neurological disorders. Lastly, I also want to acknowledge the work of our PhD student, Linus Vieira, who did most of the recordings. Thanks for your work.